basically what that diagram is showing is that the vagus nerve acts directly on the parietal cell and the G cell in the stomach. And that basically starts during the cephalic phase. So when you start to smell and sense that you're about to eat a meal, okay, that perception is transmitted to the vagus nerve, and the vagus nerve directly tells the parietal cell to start producing acid. And it does that via the transmitter acetylcholine. So the vagus nerve synapses directly onto the parietal cell. It dumps acetylcholine onto the parietal cell. The vagus nerve also synapses directly onto the G cell and tells the G cell to release gastrin, which acts locally on the parietal cell. And the gastrin also gets dumped into the bloodstream. And then the vagus also innervates the pancreas and the liver and gallbladder. And so it also is stimulating weak contractions of the gallbladder so that bile gets released into the small intestine. It's stimulating the pancreas to release enzymes and bicarbonate into the small intestine so that when chyme actually starts to enter the small intestine, it can be immediately digested, or that the digestive enzymes and bile can immediately start acting on that chyme. So basically what the vagus does is it takes those signals and kind of jump starts the process of digestion in the stomach and small intestine, so when the chyme arrives, the digestion process can start immediately. Okay, Katie, does that answer your question for the first one?
Yes.
Contribution of the vagus nerve. Okay. Hello, Macy, Jake, Caleb, fried chicken livers from KFC, gravy to dip them in. This is informal. Y'all can come up to the front, help yourselves. Please. Yes.
Can you draw the parietal cell that we had on the quiz?
Okay, we'll come back to that. So hold that thought for just a minute, the parietal cell. Okay, so then the second diagram, and so those of you that just walked in, the question was, can I explain those last three diagrams that we drew, basically what they represent? First one is the contribution of the vagus nerve to digestion. Stimulates the parietal cell, G cell, pancreas, and liver. Helps jumpstart digestion. The second diagram shows the contribution of hormones and locally released factors on digestion. Okay, so the first diagram is the nervous system. The second one is hormones and locally derived factors. And so going to the stomach, gastrin, released by the G cell, acts locally on the parietal cell. Histamine can be released locally, acts on the parietal cell, stimulates release of acid. Somatostatin can be released locally, acts on the parietal cell, inhibits acid release. Prostaglandins also can be released locally, inhibit the parietal cell to release acid. Okay, then from the small intestine, we talked about how once the chyme enters the small intestine, it's acidic from being in the stomach, it contains fat, and it's hypertonic, meaning that it has a higher concentration of salt and other components as compared to the natural environment of the lumen. And those constituents trigger the intestine to release CCK, secretin, GIP, and VIP into the bloodstream. Okay, so all of those things I just mentioned, they are released by those cells that line the villi. So remember, the villi, those finger-like projections, they extend out into the lumen and basically maximize the surface area for contact with nutrients. So the cells that line the villi include endocrine cells that release the factors that I just mentioned, but also those enterocytes that have digestive enzymes and then absorb the nutrients into the blood or lymphatic system. And then those cells that line the villi can also include goblet cells. Does anybody remember what goblet cells do?
Mucus?
Yeah. Mucus. Okay, kind of like the mucus neck cells in the stomach. So anyway, going back to the question, what that diagram represents, those factors that I mentioned that are released into the bloodstream, they act on certain cells. So VIP does what? Once it's released into the bloodstream, it does what in the blood? It's a vasodilator. It opens up the blood vessels so that more nutrients can be absorbed. VIP also acts where else? On the pancreas. So remember, if you get in a jam on the exam and you can't remember exactly what something does, there aren't that many options, right? You've got the parietal cell, you've got the pancreas, the liver, and gallbladder. So in general, if you pretty much guess that the stuff that's being released in the intestine inhibits the stomach while stimulating the pancreas and the liver, that's a pretty safe bet. Okay, so VIP also stimulates the pancreas to release enzymes. CCK inhibits the parietal cell in the stomach, and it promotes what? Bile release from the gallbladder. Secretin inhibits the parietal cell in the stomach, but promotes what? Bile production and bicarbonate release from the pancreas. CCK stimulates enzyme release from the pancreas. GIP inhibits the parietal cell in the stomach and promotes what? Insulin release from the pancreas. Okay, so if you can remember those things, remember CCK, GIP, and secretin, and you could also include VIP in that category, were the ones that we called enterogastrones, the things that are released in the intestine and inhibit the parietal cell. They inhibit gastric function while promoting pancreatic and liver and gallbladder function. Okay, is there anything that I haven't mentioned that was on that figure? I think that's pretty much it, right? Yes, Katie.
So when we're drawing the figure, when we're drawing the stomach or has parietal cells and G cells and stuff, since you didn't talk about anything being released or acted on upon the sheath and mucus cells, do we need to draw them when we draw the diagram?
Not unless you're specifically asked to. Okay, so again, like with previous exams, read instructions very carefully. Just draw exactly what you're being asked to draw. Okay, so first diagram from this last set of three that we drew, the first one is showing how the vagus nerve regulates digestive activity via its innervation of the stomach, pancreas, and gallbladder. Second diagram shows hormones and local factors. Yes, question about that?
Are the outlines going to be on the exam, or are we going to draw it?
Okay, I can't give you any more information other than to say that you have everything that you need to do well on the exam. Hey, I've given you everything that you might possibly see or encounter on the exam. Okay. Okay, then the third diagram is just the actual process of digestion. Okay, so we know that in the diet there can be fats, carbohydrates, proteins, vitamins, minerals, nucleic acids, and a few other components that can actually be broken down and used by the body. For the purpose of this class, we're just focusing on the major macronutrients: carbs, lipids, and proteins. So that third diagram basically showed if we start the process of consuming a meal, whether it be us or an animal, a pig or a dog, okay, there's prehension, the process of getting it into the mouth, mastication, coating it in saliva, and from that point on, you should be able to describe for each of those three macronutrient classes how and where does it get absorbed. Okay, and so using the diagram, you can work your way through that one at a time. So if we're talking carbohydrates, we know that in the saliva, there's salivary amylase. So starches and glycogen are getting broken down into polysaccharides, which are slightly smaller polymers of glucose. And then bicarbonate is being added to buffer that. And then we know that in the stomach, there's very little action occurring in terms of digesting the carbohydrates, the polysaccharides. They move on to the small intestine. And then what acts on the polysaccharides in the small intestine?
Pancreatic amylase.
Alpha amylase coming from the pancreas. Okay, and so then the amylase digests those polysaccharides into what?
Sucrose, lactose, and maltose.
Sucrose, lactose, and maltose, the disaccharides. So sucrose is a disaccharide of fructose and glucose. Lactose is a disaccharide of glucose and galactose. Maltose is a disaccharide of glucose and glucose. And then the last step is really easy to remember because you just take that word and change the O at the end to an A. Sucrase, maltase, and lactase, cleave those into their constituent monosaccharides. And then what you need to be really careful to remember to show is that the monosaccharide that's there at the brush border comes into the enterocyte through a transporter, and then gets delivered into the bloodstream, the portal vein, via a transporter. So remember, if we're talking about a mammal, a dog or a pig, for example, a non-ruminant, the monosaccharides and amino acids from proteins, which we'll talk about in just a second, they go to the portal vein to the liver after they leave the enterocyte. Whereas the chylomicrons from fat digestion, they go into the lymphatic system. Okay, so is everybody clear on carbohydrate digestion? Yes, Emery.
Can you repeat what you said about like after they go through the transporter into the bloodstream? Where they go after that?
Yeah, so monosaccharides, so glucose, galactose, and fructose, go into the enterocyte via a transporter, leave the enterocyte via a transporter to diffuse into the capillary bed that enters the portal vein to go to the liver. Most important thing I'm looking for is that they're entering the bloodstream. They're entering the portal vein. And then they're delivered to the liver. And then the blood from the liver drains back to the heart, back to the vena cava. Okay, so then proteins. Is there anything, we start with chewing and saliva, in the saliva that's contributing to protein digestion in general? No, not really. I mean, it's being, chewed up to maybe break it up a little bit. It's being coated with bicarbonate, buffering it a little bit, but in general, it's not being digested at that stage. Okay, so then swallowing, peristalsis in the esophagus. Okay, being sure if you're instructed to do so to label where the sphincters are at. So remember, you've got the upper esophageal sphincter, lower esophageal sphincter. Okay, enters the stomach, and then for proteins, this is really important. So this is where it's important to show that the parietal cell's releasing or producing acid. The chief cell is producing pepsinogen. The acid basically denatures the pepsinogen and converts it into pepsin. And then pepsin cleaves the proteins into smaller proteins. So in general, proteins are still proteins when they leave the stomach, but they're smaller. Okay, just like the starch in glycogen gets cleaved into smaller polymers that we call polysaccharides, proteins are cleaved into smaller proteins. Yes, Sunny?
Can you go over the functional difference between the upper and lower esophageal sphincter?
Okay, so basically when you swallow, remember that there's a voluntary phase and an involuntary phase. So voluntary is basically the action of using your tongue to propel that food or the chyme to the back of your throat. Then the involuntary part is where the upper esophageal sphincter relaxes, the epiglottis covers the trachea, the soft palate and uvula cover the nasal cavity so that nothing goes up into your nose, nothing goes down into your trachea, goes down into the esophagus. Peristalsis propels it down to the end of the esophagus, and then the lower esophageal sphincter, which you could also call the cardiac sphincter, relaxes and the chyme enters the stomach. And then remember that sphincter between the stomach and small intestine is called what? The pyloric sphincter, exactly. So those are the main ones that you would need to remember and show on this exam. Okay, any questions about that? Yes? Say that again? It's a little more complicated than that. You don't need to worry about that for this exam. And going back a second, this is kind of a strange side note. Does anybody watch the Andy Griffith Show? Okay. Have you ever seen the episode where Barney's talking about the uvula? Look that up on YouTube, please, where Barney talks about the uvula. Okay, and look at that before you take the exam, and then you'll definitely remember it for the exam when we talk about the process of swallowing. Remember that Barney Fife, uvula, Andy Griffith, YouTube. Okay. Anyway. Okay, so back to the proteins. Okay, so smaller proteins are being derived in the stomach. Remember, the mucous neck cells are releasing mucus and bicarbonate so that the acid doesn't penetrate down and burn a hole in your stomach. Okay, so remember there's also contractions that are occurring in the stomach, and the pyloric sphincter relaxes and the chyme enters the small intestine. So what's acting on the proteins in the small intestine, in the lumen, Kater?
Enterokinase.
Okay, so right, so first remember enterokinase activates trypsin. And then trypsin activates the other pancreatic proteases, including chymotrypsin and the carboxypeptidases. So the major pancreatic proteases that you need to remember for the exam are trypsin, chymotrypsin, and the carboxypeptidases. So first, enterokinase activates the pancreatic enzymes, and then once they're activated from their zymogen form into their active form, they act on those proteins and cleave them into peptides. Okay, so the only difference between a protein and a peptide is the length of the amino acid sequence. And you don't need to be concerned with what that length is, just be aware that in the small intestine, the pancreatic proteases are cleaving those proteins into smaller polymers that we call peptides. And then what's the next step? What happens to the peptides at the brush border?
Peptidates or peptidates to get amino acids and di and tripeptides.
Exactly, so there's peptidases on the brush border that cleave the peptides into amino acids and di and tripeptides. The amino acids and the di and tripeptides are transported into the enterocyte by transporters. Out of the enterocyte, transporters, and into the portal vein, bloodstream. Okay, and that's basically it. Okay, so proteins all the way down to amino acids and di and tripeptides. Digestible carbohydrates like starch and glycogen, all the way down to monosaccharides. Okay, is everybody clear on proteins? You feel comfortable with that? Okay, so then last class, or class of macronutrients that you need to be able to talk about or describe the process of digestion for lipids, mainly in the form of triglycerides. So triglyceride, it's a glycerol backbone that's esterified or connected to 3 fatty acids. So so 3 fatty acids. Okay, so basically we start in the mouth, and I haven't expected you to learn anything regarding fat digestion that starts in the mouth. As a side note, there actually is a lipase that's found in the mouth. We did not talk about that for this class. You don't need to be concerned with that. So based on what you've learned, really nothing is happening in terms of fat digestion in the mouth. Swallow, the fat enters the stomach. Is anything happening in terms of fat digestion in the stomach? Nope. Chyme enters the small intestine, and then what's the first thing that happens to the fat in the small intestine? Bile emulsifies it into small droplets. Okay, bile emulsification into droplets. Then what happens to the droplets? Pancreatic lipase has been dumped into the lumen of the duodenum. Lipase starts cleaving off fatty acids from the triglyceride. Okay, so now you have these free fatty acids in the glycerol backbone. What happens next? Right, bile turns it into mixed micelles. So you need to remember that word, micelles. And then that micelle can just enter the enterocyte. Remember the lipid bilayer membrane of the cell, it's compatible with lipids, so the chylomicron enters through the brush border membrane into the enterocyte. And then what happens in the enterocyte to the micelle? I think I said chylomicron membrane. The micelle enters the enterocyte, and what happens inside the enterocyte? Becomes a chylomicron, and then the chylomicron enters what? The lymphatic system. You got it. That's everything. Okay, any questions about that? Okay, so now really briefly, someone walk me through the alimentary canal of a chicken or a turkey, starting with the beef. Beef to esophagus, to the crop, where the feed is stored, then to the proventriculus. And what's in the proventriculus? Right, HCl gets released in the proventriculus. That's the glandular stomach of the bird. Then from the proventriculus to the gizzard, and remember there can be movement back and forth between the proventriculus and gizzard, so the acid gets mixed and the gizzard can more effectively grind up that feed. Remember, birds will ingest grit, like pebbles or eggshells, to help that process in the gizzard. Then gizzard to what? Small intestine. Duodenum, jejunum, and ileum, just like mammals. And then what's another thing that's different about a chicken and turkey? The dual ceca, exactly. Paired ceca instead of a single cecum. Hey, any questions about that? Caleb?
There's nothing, those crop proventriculus, right? There's nothing that I can do.
Exactly. From
Okay.
the crop is basically an extension of the esophagus.
Got it.
Hey, any questions about that? Okay, so one other thing, question to ask you is, what is the enteric nervous system?
It's what's in charge of peristalsis.
Okay, so it's basically independently in charge of peristalsis. What does it comprise? What is it made up of? Okay, there's stretch receptors that activate it, but what is it? What is the enteric nervous system? So in those three diagrams, we really didn't touch on the enteric nervous system. But it's really important that you understand the enteric nervous system is basically the gut's own nervous system. Remember, if you cut off the vagus nerve, if you physically go in with a pair of scissors and cut it off, the intestine will operate independently without it. It doesn't need it. Basically what the vagus does is it kind of modulates it. It can make it work faster or it can slow it down. But what physically is the enteric nervous system? That's right, there's a submucosal layer or plexus. It's just a group of nerves that are found in the submucosal layer. And then what's the other one called? Yeah, it starts with an M. The myenteric plexus. So all that the word plexus implies is that it's a group of neurons, and you've got two layers of them embedded in the wall of the intestine. And they're strategically positioned such that they surround the smooth muscle layers, which are what in the intestine?
Circular smooth muscle,
Circular
or
and
longitudinal.
longitudinal, exactly. And so by being strategically positioned around them, when those stretch receptors on the submucosal or myenteric plexus get triggered, they can cause those smooth muscle layers to contract or relax. But remember, that enteric nervous system, those two layers, those two plexuses, that can be independent of the autonomic nervous system, independent of the vagus. And what was the third smooth muscle layer that the stomach has? The oblique, in addition to the circular and longitudinal. Jessica,
Where is that one? That's just on the outside.
which one, the oblique?
Yeah.
On the inside, yeah.
On the inside?
Okay, so, yes, Katie.
So in reference to like the diagram where we drew peristalsis and like we drew that, and one of them was the bolus and then where all the stretch receptors and stuff, and the other, which one is like which kind of muscle?
Yep, so for the purpose of the exam, so Katie's question is, well, when we drew peristalsis, we showed that a stretch receptor is getting triggered when the bolus starts to move through. That chyme is causing distension and activating a stretch receptor. And the question was, say that one more time.
Which muscle layer?
Which
is
muscle layer is being stimulated to contract? For the purpose of the exam, you don't need to know that. It's much more complicated than what we depicted in class. But for the purpose of understanding the physiology, that's really all you need to know. That there's stretch receptors that are basically dendrites on neurons that represent those two plexuses in the intestine. And when they are triggered to fire an action potential, they synapse on the smooth muscle layers and stimulate those smooth muscle cells to contract or inhibit them from contracting. And that's what allows the bolus to move through the intestine, either through segmentation or peristalsis. Matt?
So the enteric nervous system is found in the submucosal layer in which other part?
Between the smooth muscle layers. So So go back to those diagrams and look at that and make sure you understand that. The two plexuses that make up the enteric nervous system and where they are with respect to those two smooth muscle layers, the longitudinal and circular smooth muscle layers. Okay, any other questions about that?
So there's two plexus as opposed of the
Yeah, so basically all the enteric nervous system is, enteric means intestinal. All that it is the nerves that make up the intestinal nervous system, and it's two groups, the submucosal and the myenteric. And they're called that because it's a layer of neurons that sit within the layer. So the submucosal sits at the submucosal layer of the intestinal wall. The myenteric, remember my implies muscle, it's between the smooth muscle layers. Any other questions? Yes.
So when we talked about peristalsis and we drew the diagram, we also talked about if a horse was galloping or when it was just relaxed sitting in the field. I'm kind of confused at how that synapses onto the enteric nervous system.
Sure. So we didn't really talk about the contribution of the sympathetic nervous system to the intestine. So remember, at any given time, for most organs, there's innervation by both the parasympathetic and the sympathetic nervous system. One exception is the adipose tissue. You don't need to know that. I only mention that because it's what I research in my lab. The adipose tissue just happens to be innervated exclusively by the sympathetic nervous system. So when your fight or flight response gets activated and you're releasing epinephrine, the fat will break down fat and release it into the blood. But in general, fat is not innervated by the parasympathetic nervous system. But most other organs, tissues are. And so the example that I gave with respect to peristalsis is when the vagus is activated. Remember, the vagus is the parasympathetic nervous system. It's one of the main parasympathetic nerves, cranial nerve number 10. When the vagus nerve is stimulated, it enhances activities that are associated with digestion. And the example that I gave is, for example, if you feed the horse some alfalfa and then all of a sudden you decide to jump on it and start galloping, basically the contribution of the autonomic nervous system is going to shift. The sympathetic nervous system has to be activated to support that activity of the animal. And basically when that happens, the sympathetic nervous system inhibits the gut. It's basically saying, okay, we're going to take this energy away from supporting peristalsis, supporting digestion, because remember, all that's taking ATP. The peristalsis requires ATP, the digestion requires ATP. Getting those sugars, amino acids, and fats into the bloodstream and lymphatic system, all of that's ATP dependent. So basically, when your sympathetic nervous system gets turned on and you have to start running, basically it inhibits that process. And that's one of the reasons it's recommended, maybe right after you eat a meal, don't go jump in the swimming pool and start swimming. You know, sit for a little while, let the parasympathetic nervous system take over and do its job. You know, let that chyme move through the intestine and get digested a little bit before you start, you know, some type of a strenuous activity. Does that answer your question, Noelle?
Yeah.
Okay, excellent. Yes.
Like, with tying into that, like, how you, like, shouldn't swim, is it, like, a true thing that, like, you shouldn't swim, like, 30 minutes after you eat? And, like, does that have anything to do with, like, how it's activating the parasympathetic versus sympathetic nervous system?
In reality, you know, it's going to differ from individual to individual. I mean, and it is basically directly related to that. It's the activation of your sympathetic versus parasympathetic nervous system. I can't really give you a better answer than that, other than it will vary from individual to individual. Okay, any other questions? So I don't have anything guided to talk about or show you. Like I said, to this point, I've given you everything in class that you might encounter on the exam. So if you're studying your diagrams, if you're looking at your slides, you know your cell types, you know your hormones, you know the terminology that I pointed to on the slide, and I said, know this, you should be in good shape. Jessica?
At one point you had mentioned that for hormones, neurotransmitters, and enzymes, you like recommended doing 5 parts first,
what you
Okay,
have.
so remember with the parietal cell, an easy way to remember that is hydrochloric acid. It's made up of a protein and chloride. Proton and chloride. So basically you just need two things in the parietal cell to make that acid. You need a way to get chloride out into the lumen. So you need a chloride channel. That's one. And then where is that proton derived from?
Proton kinase pump.
It's exactly, so it's taken out into the lumen by the proton pump, which is the target of acid reflux drugs like Prilosec. That's how they're effective. They block that pump from putting protons out into the lumen. But how is that proton generated inside the parietal cell?
Water.
Water plus
Carbon dioxide.
CO2, carbon dioxide, through the action of Ca, carbonic anhydrase, to make carbonic acid, H2CO3. The proton spontaneously dissociates from the carbonic acid and then just gets put out by the proton pump into the lumen. And that's it. You've got a chloride channel, and then you have water and carbon dioxide being made into carbonic acid, and then that proton dissociates and is pumped out by the proton pump.
On the drawing from the slides, it has like the proton that's coming like that goes out of the cell to make the acid. It has it coming like breaking off from the H2O, and the OH goes to the carbonic acid, and the H goes down to the
Yeah.
hydrogen.
So on the updated slide, I redrew that. So if you download the updated version, it is on Canvas. It's just carbon dioxide and water becoming carbonic acid. You don't need to show the dissociation of a hydroxide anion, and that, you don't need to show that.
If we
Yeah.
did, would it be wrong though?
No, it wouldn't be wrong, but you don't have to show it that way. I think it's much more straightforward and simple to show the acid dissociated. Yes.
What do histamine, gastrin, and acetylcholine do in the production of acid?
Okay, so what do histamine, acetylcholine, and gastrin do in the production of acid? So first of all, acetylcholine, where is it coming from? From the neurons, specifically from which one when we're talking about the parietal cell? From the vagus nerve. And what is the effect of acetylcholine on the parietal cell? It stimulates HCl production.
Do you have to know like specifically like what it does in the cell, or is it just like
So what did I say about the pathways for those things? What type of pathways are they, just for fun? G protein coupled receptor pathways. Did I say that you have to know those pathways? Nope. Nope. You showed me on a previous exam what a G protein coupled receptor mediated signaling cascade looks like. I don't expect you to show that again on an exam. Okay, so to answer your question, you need to show where it comes from, where the factor comes from, and the cell that it acts on, and what the output is. So for example, acetylcholine, plus sign, it stimulates the parietal cell to produce acid. Histamine is produced by cells locally in the stomach. Histamine also stimulates the parietal cell to produce acid. Gastrin also potently stimulates the parietal cell to release acid.
So in our diagram, if we have to like draw that, do we just draw like just a cell releasing histamine?
Exactly, yep. So exactly the way that we've shown it in class. It doesn't need to be any more complicated than that. A circle that represents a cell that's releasing histamine. Yes.
In class, when you talked about that, for the extracurricular question with one of those birds, I forgot what it was called. Is that gonna be like an extra question on the exam?
I won't say anything about extra credit. All I'll say is if you were here in class and you took good notes and you paid attention to me, then Matt.
Can we go over ruminant physiology that we need to know?
Okay, so quick refresher on ruminant physiology that you need to know. Okay, so what are the chambers of the ruminant stomach?
The omasum,
Okay, the rumen reticulum. Remember, the rumen reticulum basically have a fold or opening between them, so the contents move continuously between the rumen and reticulum. Remember, there are those pillars in the rumen, and basically that allows for contraction to occur and things get efficiently mixed. So rumen reticulum. Rumen is lined with what?
villi.
Papillae, which are kind of like the villi in the small intestine. And papillae maximize surface area for the absorption of what? VFAs. Remember, the primary energy source in the ruminant is the VFAs derived from bacterial fermentation of nutrients. Okay, that's why a ruminant is able to take advantage of non-digestible fibers in feedstuffs like cellulose in grass or in hay or in forages. It's because the bacteria can ferment those carbohydrates into VFAs, which are absorbed across the papillae in the rumen. And the reticulum basically will sort different sized particles, and particles that are deemed small enough to move on to the small intestine, move on to the next chamber, the omasum. The omasum, what happens in the omasum?
Water reabsorption.
Yeah, water reabsorption. Okay, it has multiple layers, so it's also sorting particles. Water's being absorbed. Then it moves on to the true glandular stomach of the ruminant, which is the abomasum. So it's rich in the same enzymes and produces acid as a non-ruminant stomach. One adaptation of the ruminant abomasum is that it's rich in lysozymes, an enzyme that you would find in your tears and in your saliva. It's basically an antimicrobial enzyme. The beauty of it in the ruminant abomasum is it lets the ruminant digest bacteria. And so a major source of protein for the ruminant is the protein that it derives from breaking down the bacteria. Okay, any questions about that? And then essentially, the chyme moves from the abomasum into the small intestine, and the rest of the intestinal tract looks pretty similar to other species. Okay, any questions about that? So going back to bacterial fermentation, so for example, bacteria will act on glucose that enters the rumen and ferment it into VFAs and what else? Gases like CO2 and methane, and also release what else? Heat, exactly. And what were the three major VFAs? Propionate, acetate, and butyrate are the three that quantitatively make up the largest proportion of VFAs in the rumen. Okay, any questions about